We are going to be producing a dilution series of solute to produce a calibration curve with which we can identify the water potential of a plant tissue, in this case potato. And this video is based on the AQA required practical number 3. We are going to be using sucrose, which is a disaccharide, and that is made up from glucose and fructose. Here we have glucose joined to fructose. You don't necessarily need to be able to draw that, but you should be aware of what sucrose is made from. We can determine the water potential of a tissue by placing samples of a tissue in a set of solutions at different dilutions. Water will then move into or out of the cells, and the sample that has no change in mass has the same water potential as the solution. Now, the chances of us getting a sample that is exactly the same water potential as the solution is very slim. So what we can do is draw a calibration curve. If we plot all of the data points that we have, work out where there would be zero change in mass, we can read across the graph to work out what the solution would be that would have the same water potential. Water potential is one of those bits where it is really good to have a very strict definition for this. It is the measure of the relative tendency of water to move from higher water potential to a lower water potential. Pure water has a water potential of 0 at 25 degrees C and 100 kilopascals. It is very important that you pay attention to the temperature and the pressure when we are looking at water potential. Here is my table 1 with the data that was given to you already filled in pink. Now, I'm going to do this based on a total volume of 20 centimetres cubed. If you're doing it the smaller volume, then obviously you can adjust, but 20 is a nice, easy number to start with. You could also do it with 10 centimetres cubed. So I'm going to go through, fill in this table, and then you can use this as a reference, or you can halve the numbers if you're going to go to 10 centimetres cubed, or if you're making up 40 centimetres cubed and sharing it around a number of groups, then you can double the numbers in this table. But it is always really, really important to make sure you get the concentration correct. Otherwise, it is going to mess up your data, mess up your graph, and give you an anomalous point on the graph, which is going to interfere with your readings. Right, everything is ready and set up for this. I've got my beaker of water, and I've got my 1 mole per decimeter cubed sucrose solution in here that I made up earlier. I have my 6 tube, which I am going to label, and we need to label them 0, 0.2, 0.4, 0.6, 0.8, and 1. Once you've labelled them, you need to complete table 1 and make up your solutions. I'm using slightly smaller volumes here. So the first one is just going to be water. I'm making up a lot less than you. So that's 2, that's 4, that's 8, that would be 12. This one would be on my table 16 centimetres cubed, but just remember I'm working at scale 10 less than you are. And then the last one over here is all sucrose. So these little pipettes that I'm using do have scales on the side, and I am keeping track of how much goes in each. Don't worry, I'm just very, very practiced at doing this. So those are my tubes and concentration: 0, 0.2, 0.4, 0.6, 0.8 and 1. I know that I haven't written it on there exactly. Now it is quite warm at the moment, so I'm actually just going to leave them in the test tube. If you've got access to a water bath, then you can put them in a water bath. I have my potato chips here all ready. What we need to do now is to dry them properly. We are not squashing and squeezing the potato chips. That's not what we're doing. We're just drying them to get the excess moisture. We're just drying them to get the excess moisture off them, so that we are not going to affect the concentration of the solutions when we put them in. So just dabbing them gently, moving them around, making sure we get each side dabbed gently. And these will be made using a small potato chipper, so that they are rectangular cuboid shape. Don't forget to get the ends. And then once we've done that, we can start weighing them. Here are my tiny pocket scales, which I guess I could take around anywhere with me. I've got the tissue paper lined up in front of here, so once I decide whether it's going to be 0, 0.2, 0.4, 0.6, I can put it in front of it so they don't get mixed up. I've got my iPad here to write the results table down, and we can get started. So that is 2.36, and that has gone down on the 0, so we can take that, just pop it in front of the 0 one over here so you don't get confused. Check that it's gone back to 0. It's always important your scales go back to 0 before you start measuring, or you're going to introduce an error into this. Initial mass for 0.2 is 1.88, and then I'm just going to line that up in front of there. Check it's gone back to 0, 1.95, line that one up. Now I know I have more here, it's so that if any are wildly different for some apparent reason, I can just not use that one. So 0.6 is going to be 2.23. Let's put that there. 0.8, 2.10. Line that up in front. And then the last one, 1.94. So we can pop that on just there. Once we've weighed them and recorded it, we can take our potato chips, drop them in to the tubes. Make sure they are fully submerged. Ah, that wasn't planned. That was just completely unscripted. Just poke that down a bit. Make sure it is fully submerged. And you can see these ones over here at the end in the 100% sucrose solution. These are actually floating a bit. If I poke it down, the same with this one. Oh, that's just a little bit stuck. These ones are definitely floating over here, so you can see a very clear difference between over here and over here. We'll start my timer, and we're just going to leave that for 20 minutes. If you've got this in a water bath, absolutely fantastic. And now will be the time to go and do some reading or sort out your folders or whatever. Once these have been incubating, we can take them out of here, dry the potato chip again, weigh it again, and then pop the results in our results table. So we don't need the solution in here anymore, so I'm just going to tip that out in there as waste. Get that out, dab it to dry it. We're not squeezing it, we're not squashing it. And then pop it on the scales. Make sure they say 0. Pop that on there and write down our results, which are 277. And then we don't actually need that anymore, so I'm just going to put that one to the side. Oh, that one came out nice and easily. Again, dab it to take off any excess, that way you're not adding any extra weight to what we're measuring. Check the balance is at 0 and write it on. Not squeezing it, just dabbing it to get all the water on the outside off. And now we've got our results, I can go and do the calculations. So you've just seen me do the experiment. Here is all of my data that I collected in the experiment. So the initial mass, measuring it before it went into incubation in the solution, final mass, measuring it after it came out and it had been dried. We can now work out the change in mass. So we are going to be doing final minus initial. So for the first one, 2.77 minus 2.36, it has gone up by 0.41. 2.15 minus 1.88, it has gone up by 2.7. 2.05 minus 1.95 equals, it has gone up by 0.1. 2.33 minus 2.23, okay, it has gone up by 0.1. Now the fact that these two are the same suggests that one of them is probably going to be an anomalous result, but we will see which one it is once we have drawn the graph. We're getting very close to no change here. 1.61 minus 1.94 equals minus 0.33. It is really important to state whether it is going up or down in the change of mass, because we are going to need that when we're drawing the graph. What we're going to be doing for percentage change, we are going to be doing change in mass over initial mass times 100. So it's the percentage change from the initial that we are doing, not from the final, not from anything else. So 0.41 divided by 2.36 times 100 equals 17.4%. 0.27 divided by 1.88 times 100 gives us 14.4. It is really important that you keep all of these numbers to the same number of decimal places. Oh, just spotted that I didn't put those on there. Same number of decimal places the whole way through. So 0.10 divided by 1.95 times 100 gives us 5.1. 0.1 divided by 2.23 times 100 gives us 4.5. So we are seeing more of a difference between these two numbers than we are between these two numbers, so that is good. 0.03 divided by 2.1 times 100, that gives us 1.4. And then 0.33 divided by 1.94 times 100, and that is 17. That is minus 17.0. And then it is these numbers that we can use to plot our graph. So here is our data from our last slide, and we are plotting sucrose concentration along the bottom here, and we're going to be plotting percentage change in mass, which is this one over here. So you can see we need to go from minus 17 up to 17.4. So what I'm going to do is to go from minus 20 up to 20 when I'm deciding what my axis is going to be. So now I've worked out the axis on the right hand side, I can start to plot my points. So 0 was 17.4, 0, 15, 16, 17.4, so that's going to go there. 0.2 was 14.4, so 0.2, 10, 11, 12, 13, 14. 0.4, 0.4 was 5.1, so quite a sharp drop here. Be interesting to see what the graph looks like. So that was 5.1. 0.6 was 4.5. 0.6, 0, 1, 2, 3, 4.5, as I expected. It looks like that's going to be an enormous result. 0.8 was 1.4, and then 0.1 was minus 17. So with these, we do need to put in a line of best fits. The best way to do this is to get a ruler, a clear ruler, or if you're lucky enough to have your hands on a line of best fit ruler, then a line of best fit ruler. Put that on the bit of paper and draw your line through that. I'm going to draw my line here, like this. You can see that there's an even distribution of points either side of it. Most of the points are pretty close to the line, with a couple of points a little bit further away from the line, but they're evenly further away from the line. If I drew it, for example, down here, these points would be really far away. Even if I joined up these 3 points, I'd probably get a pretty straight line, but then all of these points would be way over the other side. Now what we need to do now is find where 0 would be. So we put our ruler against 0% change in mass, and whenever you do this, please always draw your lines, because the examiners are expecting to see your lines on there, and they are looking for that. So we can see that this has come just past here, so that would be about 0.62 moles per decimeter cubed. Now if you have a different value, do not worry, it's going to be changing based on the type of potato you have, how old the potato was, lots of different things like that. In the exam, it is unlikely you'll be asked about exactly this practical, so you need to know ways we can change it ever so slightly, some alternatives, some improvements even that could be made to this practical. So if you get asked something slightly different in the exam, it doesn't throw you off. It is basically the same practical, but with something ever so slightly different to make it a different practical, so that you are using things, you're applying your knowledge, you're using things out of context, and there's going to be something slightly different that makes you actually think instead of just recall things that you learned. This is a higher level skill. So instead of using sucrose solution, we could have used sodium chloride as the solution. This would have had exactly the same method, exactly the same results analysis, but it would have been a different practical. We could have used a different vegetables such as carrots or parsnips or Swedes. There are lots of different ways we could change this to make it a different practical. We could vary the surface area of the potato. You could change the time that it is left in solution, or even the temperature that it is at. You could change the range of solutions or the interval of solutions that you use. Instead of doing a stepwise dilution, we could have done a serial dilution. So you'd get a set of different dilutions going from 1.5, halving it down to 0.75, halving it again down to 3.8. This will give you a different looking graph. Now, while your graph may have been absolutely perfect, you saw from my graph that not all the points were on the line exactly. So there was some error in my experiments. Now, we need to know about the different types of errors and how they would impact the results. So an error could be not drying the potato fully when it went into the solution initially. You'd then be adding in water to the solution and changing the concentration of the solution very slightly, but it would still be changed. If you accidentally left some of the skin on the potato, if you were cutting it yourself, then the skin would have a different permeability to the cells and different volumes of water would go in and out. If you didn't dry the potato fully before weighing it, then that would have added added to the mass of the potato when you put it on the scales, so the final mass wouldn't have been accurate. This will lead to an increased mass and an increased change in mass. And then finally, if you made the solutions yourself, you might not have made the solutions correctly, which would give you an anomalous result. Ouch! This is why in some videos I have unexplained scratches.